The brainstem is a vital part of the central nervous system that regulates bodily functions. In this video, we will explore the anatomy, functions, and clinical significance of the brainstem, including its organization, neurotransmitter systems, cranial nerve nuclei, autonomic functions, and more. Let's delve into the intriguing world of brainstem histology. The brainstem connects the spinal cord to the brain and includes the medulla oblongata, pons, and midbrain. The medulla controls vital functions like respiration and blood pressure. The pons relays information and controls facial movements and sensory processes. The midbrain is responsible for motor and sensory functions, visual and auditory processing, and coordination. Understanding the brainstem's organization is important in comprehending its functions. In the brainstem, various neurotransmitter systems, such as serotonergic, dopaminergic, noradrenergic, and cholinergic systems, are essential for mediating different brain functions. These systems regulate important processes like mood, sleep, reward, attention, and memory. Knowledge about these systems helps understand brainstem-related diseases and develop treatments. The brainstem contains important nuclei that control various functions of the head and neck. These nuclei are located in specific areas and are connected to other structures in the brain. Examples include the oculomotor nucleus for eye movements, trigeminal nucleus for facial sensation, and vagus nucleus for controlling organ functions. Knowledge of these connections is crucial for diagnosing and treating cranial nerve disorders. The brainstem plays a crucial role in regulating basic autonomic functions related to cardiovascular, respiratory, and gastrointestinal systems. The medulla, in particular, houses various nuclei responsible for controlling heart rate, blood pressure, and respiration. The nucleus tractus solitarius receives sensory information related to blood pressure and initiates appropriate autonomic responses to maintain homeostasis. Additionally, the ventrolateral medulla contains the rostral ventrolateral medulla, a critical center involved in sympathetic control of blood pressure. Dysfunctions in these brainstem regions can lead to autonomic dysregulation and contribute to the development of conditions such as hypertension and autonomic disorders. The brainstem regulates respiration through its respiratory centers, such as the dorsal and ventral respiratory groups in the medulla. These centers receive input from chemoreceptors and lung stretch receptors. The brainstem also integrates signals from the pons and cerebral cortex to allow for voluntary control of breathing. This knowledge is crucial for diagnosing and treating respiratory disorders. The medulla oblongata houses important centers for cardiovascular regulation, including the cardiac and vasomotor centers. The cardiac center, located in the medulla's lateral part, controls heart rate and strength of contraction. It receives input from various sources, including baroreceptors and chemoreceptors, and adjusts cardiac output accordingly. The vasomotor center, situated in the medulla's ventral part, regulates blood vessel diameter, blood pressure, and overall vascular tone. Dysfunctions in these medullary centers can contribute to conditions such as arrhythmias, hypertension, and heart failure. Lesions in the brainstem can result in various cranial nerve disorders, leading to a wide range of symptoms and clinical presentations. Depending on the location and extent of the lesion, cranial nerve deficits can manifest as facial weakness or paralysis, impaired eye movements, or loss of sensation in specific areas of the face. For example, lesions affecting the facial nerve nucleus or its fibers in the brainstem can result in facial weakness or inability to close one's eye. Identifying and localizing brainstem lesions associated with cranial nerve disorders is critical for accurate diagnosis and appropriate management. The brainstem contains central pattern generators, CPGs, which are neural circuits responsible for generating rhythmic movements such as walking, running, and chewing. These CPGs are found in various regions of the brainstem, including the medulla, pons, and midbrain. They receive input from higher brain regions and sensory feedback to coordinate and modulate motor activity. Dysfunction in these locomotion control centers can lead to gait abnormalities, movement disorders, and difficulties in performing coordinated movements. The brainstem is involved in several important reflexes that help protect and regulate the body. These reflexes include the pupillary reflex, which controls the size of the pupils in response to light, and the gag reflex, which safeguards the airway from foreign objects. Other brainstem reflexes include the corneal reflex, vestibulo-ocular reflex, and cough reflex. These reflexes can be assessed clinically as part of a neurological examination and provide valuable information regarding brainstem function and potential pathology. The vestibular system, located within the inner ear, plays a crucial role in balance, spatial orientation, and coordination of eye movements. It communicates with the brainstem through the vestibular nerve and relays information to various brainstem nuclei, including the vestibular nuclei. These nuclei integrate vestibular signals with visual and proprioceptive inputs to maintain equilibrium and coordinate appropriate motor responses. Dysfunction of the vestibular system or its connections with the brainstem can result in vertigo, dizziness, and balance disturbances. The brainstem plays an integral role in regulating the sleep-wake cycle in various stages of sleep. The reticular formation, spanning throughout the brainstem, contains nuclei responsible for arousal, alertness, and the transition between sleep and wakefulness. The ascending reticular activating system, ARAS, sends projections to the thalamus and cerebral cortex, promoting wakefulness. Conversely, the ventrolateral preoptic nucleus inhibits the ARAS, promoting sleep. Dysregulation of these brainstem structures and their connections can lead to sleep disturbances and disorders such as insomnia, sleep apnea, and narcolepsy. During embryonic development, various genetic and environmental factors can lead to abnormalities and anomalies of the brainstem. These abnormalities can range from minor structural variations to severe malformations that may significantly impact neurological function. Examples of developmental abnormalities of the brainstem include Chiari malformations, where part of the cerebellum herniates through the foramen magnum, and Dandy-Walker syndrome, characterized by cerebellar and brainstem hypoplasia. Understanding these developmental abnormalities is crucial for accurate diagnosis, prognosis determination, and appropriate management strategies. A thorough understanding of brainstem histology is essential for medical students to comprehend its complex anatomy, functioning, and clinical significance. Topics such as neurotransmitter systems, cranial nerve nuclei, and autonomic functions provide valuable insights for accurate diagnosis and management of brainstem-related conditions.